thank you all for logging in. It's a real pleasure to have been asked to give one of these talks. I'm going to talk today about work in the lab over the past number of years that has aimed at trying to understand some of the neural circuitry that is controlling aggressive behavior. And more specifically, we're quite interested in understanding what drives the motivation of an individual to engage in aggressive behavior. And so here's my pulsatory. Oh, a little bit of feedback there if anybody hasn't muted, but aggression obviously is a complex behavior because it's something that is advantageous, evolutionarily speaking, in order to procure resources. In our society and in humans, but not just humans, in most organisms, aggression is a way in which they can ensure that they have enough food and resources and to protect their mates and their families from other aggressive individuals within the species. But it also can become maladaptive in many situations, and in the context of psychiatric illness, can lead to a whole host of stress-related disorders that we're all studying, such as depression and anxiety. In large part, and to simplify things, we oftentimes think of aggression in two ways, although this is super simple. So it's a continuum, and it I think kind of distills it down to the two major aspects of aggressive behavior. One in which it's proactive in nature. This is what we think of as being kind of premeditated and goal-directed. You know, one way to think about this is Norman Bates or other psychopathic behaviors where aggression is actually planned out, and it's planned out and may actually be something that is rewarded in that individual. Aggression can also be reactive in nature, and so this oftentimes is retaliatory or defensive. One example of this is territorial aggression, where an individual within the species is really trying to defend its resources amongst another aggressive, threatening animal, and so they engage in this type of reactive, retaliatory, defensive behavior. We know a lot about territorial defense, both I think at the neurobiological level and also kind of at the ethological level. I think we know a lot less about proactive, premeditated, and goal-directed aggression. Over the years, there hasn't been as much investigation into the mechanisms that drive this form of aggression. And so we really set out to understand what it is that drives aggression in certain individuals, and what it is that drives this form of proactive aggression. And so we got some insights early on from work in humans. These were done in the scanner, in the fMRI scanner, where individuals were performing some type of tasks, and they were collecting real-time information about blood oxygen utilization throughout their brain. The study I'm highlighting here was done in aggressive children with aggressive conduct disorder. Aggressive conduct disorder is oftentimes a prequel to antisocial personality disorder or psychopathy. The similar results have been obtained in those adult-only diseases. And so in this study, what they did was they, as I mentioned, put the kids in a scanner, and they showed them pictures, some of which were, see if I can get a pointer here, some of which were quite benign, right? So this is an example of a person just placing their hand on the back of the trunk of their car, versus one in which they were witnessing an individual getting hurt. And so then in this case, the individual was slamming their hand in a car door. And what they noticed was these kids with aggressive conduct disorder had some interesting patterns of activity throughout their brain reward circuit. In particular, when they witnessed images or viewed images of individuals that were harming themselves or being harmed by others, they saw activation of the ventral striatum or nucleus accumbens. And this was quite interesting to us, having studied for years the nucleus accumbens and its role for mediating a whole host of rewarding activities, including drugs of abuse, we thought this was possibly an indication that these individuals were exhibiting this, what they were calling atypical empathic responses, and what we were calling reward to witnessing others being harmed. So how do we get at this in an animal model, and can we test circuits and functional questions about whether or not certain individuals find aggression to be rewarding, or harm to others to be rewarding? So we went to a resident intruder model. We had been using this over the past 10 years or so to understand how stress impacts brain circuits involved potentially in depression and anxiety. And so we just modified the protocol in order to study the aggressive interaction itself, rather than to study the consequences of such an aggressive interaction. And the way we do this is we take a large outbred CD1 mouse. These are genetically quite diverse, and that's actually quite important in terms of maintaining an aggressive phenotype. Over the years, we've selected these inbred mice so that they aren't aggressive towards us, and in doing so, we've selectively bred out a number of the important genes that are involved in aggressive behavior. And so we select these outbred mice, and we introduce an inbred subordinate intruder into their home cage, and then measure the physical altercation that ensues afterwards. So in these initial studies, we used exclusively male mice. And so for the first 2/3 of my talk, I'm going to only show data from male mice. Part of this is that female mice oftentimes exhibit far less territorial, overt physical aggression towards intruders. And it's taken us quite a while to reestablish ethologically relevant models for female aggression, which we've just established in the lab, but have not yet been able to collect important biological information on what drives their forms of territorial aggression. And so in our model, we essentially introduced this animal over a series of 3 days to get a basic aggression profile. We want to know how often they attack, how long they're attacking, and what types of attacks they're engaging in towards these intruders. And this is what we see across the population. Each point represents an individual animal that we screened in that resident intruder task in the previous slide. And you can see that approximately 70% of all mice that we screened engaged in some form of aggressive behavior towards the intruder. 30%, or a little bit less than 30%, did not attack during any of the sessions of the resident intruder. We call these animals non-aggressive. Now, it's interesting to note that within this non-aggressive population, you can push them a bit more. So you can extend the resident intruder task, you can provoke them. And if you do those types of environmental manipulations, you can get another, I would say 15% to convert over and engage in aggressive behavior. But there is this subpopulation of individuals that simply do not attack throughout any of the sessions that we've witnessed. But if you look at the animals that do attack, again, you can see this wide distribution of aggressive behavior. This is showing just the latency to attack. This measure correlates perfectly with other measures of attack behavior as well. On the one hand, you've got animals down here that attack really quickly. These animals also attack quite intensely and for long durations. And it's interesting to look at the, qualitatively speaking, the type of attacks that this population engages in is quite different than the type of attacks that the rest of the population engages in. They often go for kill areas, for lack of a better word. We see them going after the jugular vein, we see them going for the underbelly, whereas a typical aggressive interaction with an intruder, which we view as to be more territorial in nature, is to bite on the base of the tail, almost as if these animals are telling the animal to get away from them and leave them alone. Whereas these ones, qualitatively speaking, were engaging in what we consider more of a proactive phenotype. They were going after these animals and they seemed to enjoy it. The other thing that we noticed was that these animals over time became more and more aggressive. The more you expose them to opportunities in which they can engage in aggression, the more these animals the longer they attacked, the quicker they attacked, and the more they converted towards attacking in places that seemed to be meant to harm rather than to just push the animal aside. So ultimately what we wanted to do in the lab after seeing these profiles was to test whether or not this behavior itself was somehow rewarded or reinforced, and if that was what was kind of driving this type of proactive aggression that we see in our highly aggressive animals. And so what we did was we actually selected mice from this upper quartile of most aggressive animals here, as well as the lower quartile of non-aggressive animals. And we then put them through a standard condition place preference assay, which we've used, and I know a number of other labs here at Sinai have used to measure drug reward. We just replaced the drug of abuse with a small intruder mouse that they were now allowed to attack. We videotaped, so we made sure that the attack ensued, and we also made sure that the aggressive experimental mouse never was attacked by the other individual. Yes.
Scott?
Yeah.
Scott, this is Andy Chess. I have a question. Why did you say that you think that the animals are enjoying it? You said that some of the ones that have a short latency you think are enjoying attacking.
That's what these tests are meant to get at, and so I'll get into that in a few seconds. Give me 2 slides to get at that question. They were qualitatively speaking. Well, let me get at because we have actual data that shows that these events are actually quite rewarding and reinforcing to them. So let me just jump right to the chase. So here's the data that proves at least that the salience of this interaction was rewarding to aggressive individuals. So following this conditioning session where they were allowed to attack animals in this particular environment, we then placed the animal back into the CPP chamber with free access in the absence of an animal. We simply asked which environment did they prefer to spend their time in. And they always spent their time in the chamber in which they were allowed to attack if they were aggressive at the onset, whereas non-aggressive animals found these intruder interactions to be quite aversive and avoided them. And this is the quantified data right here. So this suggested that there was some type of conditioned reward effect in these highly aggressive animals, again, which we interpret as it being rewarding to these individuals. But I think the best data to really prove this is a video. And to really convince you all that this is not simply territorial aggression, but this is actually proactive in nature, it's planned and reinforcing. So this is a model that was developed by Sam Golden, who was a graduate student in my lab at the time, who had developed the initial condition place preference assay as well. And in this model, he's essentially, what he's doing is training animals to engage in a second order conditioning task in order to receive access to an intruder mouse in which he can attack. And so in this chamber, you'll see a light come on right here, and that cues a lever that the animal can then press. The trap door opens, the animal gets pushed in, and this is the premeditated, proactive part. They do this all the time. They guard the door and they make sure that it closes so that the animal cannot escape before they can go and attack. And you can see that right there. So the animal comes out, session 2 goes forward, and they do the exact same series of behaviors. So effectively, the ability to attack and subordinate another individual elicits a conditioned reward response. It's reinforced, and it's planned out and proactive in nature. Andy, did that answer your question?
Yes, thank you.
Okay. So you can see him guarding the door again. Just striking that these animals have that level of interest in attacking another individual. So of course, this to us pointed to circuits in the brain that we already had been studying to be involved in other aspects of reward behavior. This is just showing a simplified cartoon of some of the various hotspots that we know are involved in both natural and drug rewards. And what we wanted to do initially, this was probably prior to the days of whole brain C-FOS mapping, so we did it the old-fashioned way. We took animals, we had them engage in an aggressive interaction. We then sacrificed them immediately afterwards, and stained for C-FOS through the entire extent of the brain, focusing in on areas that we know are involved in reward. And I'm not going to show you all the data. I'm just going to go straight to one of the areas of the brain that we thought to be quite interesting. This was the lateral habenula. An interesting side note, this was before Paul Kenny joined us, but he was giving a talk on the role of the lateral habenula in nicotine addiction the day that Sam was analyzing these, which prompted him to go into the microscope room and immediately look at the lateral habenula and saw this very interesting differential pattern of C-FOS expression in the lateral habenula, which was suggestive of differences in activity of nerve cells within that region.
I'm surprised I'm not a co-author of the paper then, Scott. That's very
We did acknowledge
to
you,
hear,
Paul,
you know?
You were our muse, so we did acknowledge you. So the lateral habenula is really interesting. As I mentioned, Paul's been studying it for its role in other forms of addiction. And this is a nice review from Garrett Stuber's lab defining what the input-output structure of the lateral and medial habenula is, but also highlighting one of its well-established prominent roles in mediating aversion. So in this study that's depicted by this cartoon, they did what's called a real-time place preference assay using optogenetics. And in this, they expressed channel rhodopsin directly into the lateral habenula. They put a blue fiber optic cable to deliver blue light directly into the habenula to activate those cells. And whenever the animal walks into one of the two conditioning chambers, the blue light comes on, activating the habenula, and the animal leaves that side of the chamber, and it elicits a real-time place aversion. So in large part, this is consistent with a lot of other literature, both in rodent and primate brain, which argues that the lateral habenula is critical in sensing a reward and aversion, and that activation of the habenula leads to aversion. So based on this, and based upon our C-FOS excuse me, profile, this pattern would argue that non-aggressors have a more active habenula, maybe explaining why they find aggressive interaction to be aversive, and aggressors have a less active habenula, possibly explaining why they find it to be more rewarding. So we wanted to really know whether or not the habenula was encoding valence during these social interactions, both good and bad, right? If you remember, the non-aggressors showed an equal but opposite change in valence in response to an interaction like this. And so the way we did this experiment was to inject a calcium sensor directly into the habenula. This is GCAMP6S. We did it non-conditionally, and I'll explain to you in a few minutes why we did it in the original study. But we did a non-conditional expression of GCAMP, calcium indicator, to detect neural activity in vivo in real time. We then allowed the virus to express for a period of time, and then conditioned animals for aggression. Well, we conditioned them with an intruder mouse in this standard CPP assay, as I had explained previously. We then put the animals back into the chamber during the test phase and measured calcium activity as they traversed through this intruder paired and intruder unpaired side. So the side in which they were able to attack versus the side that they were not. And this is what we found. In aggressive mice, as they traversed into the side in which they were allowed to attack, their lateral habenula quieted down relative to the activity patterns that we saw when they were in the alternate chamber. This is just showing the average peak per minute rate of calcium transients in paired versus unpaired in aggressive mice. We found exactly the opposite in non-aggressive mice. So their lateral habenula became more active as they traversed into the side they were paired with a social target, and that's quantified here. It trended towards significance and was probably just low powered, but you can see that's quite a large difference between their activity patterns when they were in the paired versus unpaired chamber. I'd like to give a big shout out to Erin Calipari, who at the time was in Eric Nestler's lab, who helped us to set up this in vivo calcium assay fiber photometry. She's now at Vanderbilt. So to test functionality, we then injected channel rhodopsin, again non-conditionally, into the LHB. conditioned animals for with the intruders as we did previously, and then stimulated during the expression phase of the test, right? So this was transient stimulations throughout the entire expression phase. So the animals naturally acquired place conditioning, but the habenula was modulated only during the expression of the behavior. And you can see that in an aggressive animal that would normally show CPP, this is a control condition, when their lateral habenula was stimulated, they actually showed conditioned place aversion, similar to non-aggressors. So this suggests that the lateral habenula, at least as a whole brain circuit, can encode environmental information about aggressive social interactions and non-aggressive social interactions, but that it is functionally related to the expression of valence in these assays. So at the time, we were kind of thinking about the habenula quite simply. In large part, the habenula had been characterized as an excitatory hub for the brain reward circuit, composed exclusively of excitatory glutamatergic cells that receive a lot of innervation from regions throughout the brain reward circuit that we know regulate reward and aversion. And so Megan Flanagan, a grad student in the lab at the time, now at UNC with Tom Cash, wanted to study some of those upstream regulators. And she began to probe various systems throughout the brain to see what might look interesting. And I'm not, in the interest of time, I'm not going to show you all the circuits that she probed, but I just want to focus in on one that we thought was particularly interesting. She found a previously uncharacterized, but now more and more people are starting to appreciate this circuit, containing orexin neuropeptides coming from the lateral hypothalamus. This is showing in the left panel fibers that are orexin positive, presumably again coming from the lateral hypothalamus, since this is the only source of orexin in the brain. We did retrograde tracing to confirm that orexin positive cells in the lateral hypothalamus were indeed projecting directly into the lateral hypothalamus. And PCR results suggested that following an aggressive interaction, aggressive mice had higher expression of orexin receptor 2 directly in the lateral hypothalamus, directly in the lateral habenula. We obtained an orexin-Cre line to test causal hypotheses about the role of this orexin circuit. And so to perform these studies, we injected a Cre-dependent channel rhodopsin or halorhodopsin directly into the LH of orexin-Cre mice. We then placed our fiber optic cable directly in the lateral habenula so that we could either stimulate or inhibit terminals from the lateral hypothalamus into the lateral habenula. And I just want to show you a video. These were aggressive mice. This is the same aggressive mouse actually with light off and light on situation. In this case, we're actually inhibiting the inputs. So we're reducing orexin signaling from the LH to the LHB. And you'll see how that affects aggressive behavior. Turn my laser pointer off. So this is the mouse that's now exposed to an intruder. He's attacking normally, but when we turn the yellow laser on, this same mouse does not elicit an attack during the entire session. So you can see this input strongly regulates some aspect of aggression initiation. And we think that it's possibly changing the motivational valence of such an interaction, thereby inhibiting the animal's drive to engage in it. So this is quantified here on the left. You can see animals where their inputs to the lateral habenula were inhibited show dramatically reduced attack duration. They also show aversion to the chamber in which they were conditioned with a social target, suggesting that this input regulates both aggression intensity as well as the valence of such interactions. we were able to do exactly the opposite by using channel rhodopsin stimulation of these inputs. And so we were actually quite confused by this data. This was not what we expected. Effectively, orexin receptor 2 is largely described as coupling to a GQ receptor, and then should, in theory, cause activation of the downstream neuron. And I mentioned earlier, the literature had told us that really the LHB was an excitatory hub, that there were no inhibitory neurons in the LHB, and that they only contained these glutamatergic projection cells. And so in order for this to make sense, which it didn't, when we inhibit this synapse, it should have decreased aversion and increased aggression based upon what we had shown previously in our data with non-conditional stimulation of the lateral habenula. So we thought about this for a while, and we were thinking about maybe non-canonical pathways of orexin signaling. We came across an Friedman Brain Institute in situ hybridization study showing that there was a tiny pocket of GAD2 mRNA expressed in a very defined region of the lateral habenula. And so we confirmed this in our own hands, and indeed there is a small population of GAD2 containing lateral habenula neurons. GAD2, of course, is a marker of GABAergic neurons in the brain. But it is, as others had said, largely excitatory. Most of the cells here are excitatory, and they contain VGlut2. So with the help of Beth Lucas and Roger Clem, we were able to show functionally that these nerve cells could support inhibitory tone that was mediated by GABA directly in the LHB. So to do this study, we expressed Cre-dependent channel rhodopsin into the LHB of GAD2 Cre mice, so targeting specifically those GAD2 cells. We then stimulated them in slice, measuring neighboring cells, measuring inhibitory currents evoked in neighboring cells as we light stimulated those GAD neurons, and that's shown right here. What's interesting is that picrotoxin, which is a GABA-A antagonist, was able to fully block these, confirming that these are indeed signaling through GABA. When you look at the LHB as a whole, again, in our initial study, we looked just at total FOS-positive cells following aggressive interaction, and this was the profile we got. If we broke those down into GAD2 versus VGlut2 cells, you can see it's a very interesting pattern. The VGlut2, which is the predominant cell type in the LHB, follows the pattern that we saw initially, whereas GAD2 cells show exactly the opposite. These cells seem to be activated in response to aggressive interaction in aggressive mice. And importantly, if we functionally manipulate these cells, we can regulate aggression and aggression reward. So again, for this study, we injected a Cre-dependent channel rhodopsin directly into the GAD2 Cre mouse line in their LHB. We then light stimulated this pocket of GAD2 cells. We were able to moderately increase attack duration and very significantly increase the preference for the chamber in which they were previously allowed to attack, suggesting that these cells really do drive aggressive behavior, but they drive it in a way that's quite different than the glutamatergic cells that we had studied previously. These cells, importantly, respond to orexin. This was work done with the help of Bridget Matakainen and George Huntley's lab before she moved on to NDS, I believe. And what she did here was, again, using the GAD2 Cre line, we labeled those cells with GFP, and then she bath applied orexin peptide directly onto those slices and measured firing rates of those cells in response to orexin. And you can see that they increase their firing rate once orexin is bath applied. And then last but not least, we showed that signaling through the orexin receptor 2 was critical for the expression of this phenotype. So for this, Hossein Aliassane in my lab who now is with the CEYE program, so many of you I'm sure know him, had generated or helped us to design a mirror to knock down OXR2 receptor. We then package this into a DIO Cre-dependent AAV, and again inject it into the GAD-Cre LHB, and we were able to both increase attack latency, decrease attack duration, and reduce aggression reward as shown over here. We could also systemically administer a pharmacological antagonist for OXR2, and this dramatically reduced aggression and aggression CPP. One of the issues with this strategy clinically is that other centers in the brain that are very well characterized have shown that orexin 2 receptor antagonists cause broad changes in sleep-wake cycle patterns. And so high doses of systemic OXR2 antagonists can promote sleep, which is an undesired side effect. We tried to dose this in our animal studies such that we found no evidence of alterations in sleep-wake or general motor activity or arousal. But again, this is a concern for treatments that would operate on blocking OXR2. But again, our in vivo data shows that the lateral habenula, at least, is a site of action of orexin, and it goes through the orexin receptor to mediate these aspects of aggression. So this is our newly proposed circuitry. We've identified a previously uncharacterized GABAergic neuron and shown it to be functionally able or capable to provide inhibitory tone onto glutamatergic cells that we know control aspects of aggression and aggression reward. It does so at least in part through release of orexin peptide coming from the lateral hypothalamus that then seems to activate these GABAergic neurons to then silence the glutamatergic cells. We don't know this for certain. We don't think it's synaptically connected. This is based on some rabies studies that we did in our lab. We think orexin is probably communicating with GABA cells at extrasynaptic sites or through something akin to volume transmission. We found no evidence of direct synaptic connectivity between these two cells, though. So at this point, I want to transition, because really what the lab is interested in now is understanding negative social interactions and antagonistic social interactions, for example. So we use the social defeat stress model to understand a lot of the consequences of social stress. In the first half of the talk, I told you about some of our deeper understanding of the causes of social subordination. Now I want to talk about some of the consequences. So what happens in the individual that's receiving this form of aggressive social subordination? There's some interesting studies out from Anna Weinberg's lab recently showing that these types of social subordinations or bullying in kids and adolescents and even in adults can cause really dramatic effects to social behavior. And in particular, it seems to blunt neural responses to rewards, including social rewards. So many of us find social interaction to be quite rewarding. And what we know in the case of social subordination or social stress, individuals that are experiencing this don't seem to find social interaction to be rewarding anymore. And this is associated with things like reduced activation of the nucleus accumbens, for example. So again, we wanted to come back to the social defeat stress model that we had used previously. This is all unpublished data, and it's been led by Long Lee, a postdoc in my lab. The social defeat stress model classically goes like this. We take an aggressive CD1 mouse, the one I've been telling you all about for the past 30 minutes. We then expose an intruder, a C57 inbred intruder, to his home cage or her home cage. We've now developed a model for females. 5 to 10 minutes of aggressive social encounter ensues over the next 10 days. And then in between those aggressive encounters, the animal's housed for 24 hours with this form of sensory contact that we know is also a stressor. At the end of the 10 days, we determine resilience versus susceptibility based upon social interaction with a CD1 target mouse, and that's shown right here. Resilient mice interact quite normally with this CD1 mouse. They do not avoid, whereas susceptible mice tend to stay in the corners of the chamber and avoid completely the target zone whenever the CD1 mouse is available. And we've interpreted this oftentimes as a measure of social reward deficits. Although over the years, we've received criticism, right? So people have questioned whether this animal is truly resilient. Maybe they're just stupid. Why are they going to interact with a CD1 mouse when this mouse or a mouse like it had been beating it up for the past 10 days? Others have argued that this is not reflective in any way or shape or form of social reward deficits, but rather this is reflective in the susceptible mouse of fear and trauma avoidance. And so we really wanted to remove this potential confound of what social interaction measures were actually explaining or depicting in the animal's behavior. And so to do this, we got rid of the CD1 as a social target here, and we replaced him with a same-sex, same strain juvenile mouse. And so you can see the cartoon here in the CPP chamber. These juvenile mice are not at all aggressive, obviously. They're about a third the size of the adult. They're the same, again, the same strain and the same sex. So they're completely non-threatening. And under normal situations, they can induce a conditioned place preference. We do our conditioned place preference assays identically to how we do our aggression conditioned place preference assays, except we obviously use a different social target. And what you can see here is that both male and female mice elicit a conditioned place preference to the juvenile, and that's shown here. The paired side is the side that they were exposed to the juvenile. And after 4 days of conditioning, both animals, both sexes seem to prefer to spend time in that side of the chamber. However, if you look at socially stressed mice, I've got this now broken down to unstressed, resilient, and susceptible mice. You can see susceptible mice fail to develop this conditioned place preference for the juvenile. They don't seem to find this social interaction to be all that rewarding, which is similar to data that has been shown in humans. This data, I believe, was from females, but we see identical results in males as well. So coming back now, we have a little bit of a better tool to map C-FOS throughout the brain. I think Paul discussed this technique in his FBI Cyber Series talk, but this is a method whereby we can completely clear the brain and make it transparent, as shown here, and then stain C-FOS puncta throughout the entire extent of the brain. And you can see quite nice resolution of the individual C-FOS puncta throughout the entire extent of the brain. We then took mice following social defeat stress. We exposed susceptible and resilient mice to a juvenile intruder and then sacrificed them an hour and a half later so that we could stain for C-FOS throughout the entire extent of the brain. And the table here shows some of the interesting effects that we found. So looking across, this is just a few of the areas that we found to be quite interesting. You can see some of the same players that we've defined previously. Not surprising, amygdala structures were coming up, areas of the thalamus and subiculum. The entorhinal cortex was really strongly affected, as was the lateral septum. And so we really had started our initial investigation into areas that were extremely strongly regulated. And so the lateral septum was what we decided to follow up on in more detail. The lateral septum is an interesting structure. It's located really between between the ventricle here, the lateral ventricle. It's very close to a number of interesting reward centers, but it also receives a lot of input and sends input out to a number of areas that we know are involved in various aspects of social behavior, as well as those involved in social defeat stress. And so as we started to probe through the literature, we noted that the septum plays a large role in a lot of behaviors that are important for mood and emotion, including aggression. Many of you may have heard of a situation or a phenomenon called septal rage. This is where an individual has a portion of the septum ablated, and the individual then becomes extremely aggressive and violent. But the septum is quite diverse. It has a number of different cell types. It's got a very complex anatomy. It's quite a large structure, sitting just adjacent to the midbrain, taking up quite a lot of real estate here. And so it can also play roles in pro-social reward and pro-social behavior, as well as responding to stress and threats. And so this seemed like kind of the perfect target to really go after and better understand our findings on social reward deficits and susceptibility. So a further dive into the types of cells that may be activated based upon their neurochemical profiles showed us that it was a population of neurotensin positive GABAergic neurons that lie just adjacent to the ventricle here. You can see this shown down here. So when animals following social defeat stress, when mice were exposed to a juvenile, the susceptible mice showed a very strong activation based on C-FOS in these neurotensin positive GABA neurons in the lateral septum right here. So we did a bunch of other studies to test whether neurotensin itself was being regulated and found no evidence that it was, but we're using neurotensin as a marker of a cell type rather than studying the role of neurotensin itself. So for subsequent studies, I'll be referring to this as LSNT. And really what we're trying to understand is how GABAergic tone elicited by these neurotensin cells in the septum are mediating aspects of social reward. I do want to mention, though, that if we started to look through the literature, and one of the interesting findings that we came across was that acute and chronic stressors really strongly activate these cells, as well as others in the lateral septum, as do threats in the environment. So it's almost as if these susceptible mice are experiencing a juvenile, and that their lateral septum is telling them that it's a threat, that they're inappropriately assessing the juvenile social target as being threatening, which may occlude social reward processing. It's a hypothesis that we'd like to test in the future. So coming back to our real-time measures, we wanted to understand what's happening during this juvenile SI test. What is it that the lateral septal neurotensin neurons are doing during the test? And is it somehow associated with some form of social interaction? So we came back, we used, again, the same strategy that I had used in our previous slides. We used a Cre-dependent GCAMP-S. We injected it directly into the lateral septum of neurotensin Cre mice, and the expression profile is shown here. We put these animals through 10 days of chronic social defeat stress. We define them as susceptible or resilient based upon their avoidance of a CD1 target. We then take these mice, expose them to a juvenile in their home cage while measuring calcium activity from those neurotensin cells. We did this again both in males and females following defeat, and what we found was quite interesting. So the upper panel here of C is the behavior. So this is measuring the time that the animals spend either during social investigation, where nose-to-nose contact is occurring, versus avoidance, which we measure as an event where a juvenile mouse approaches another animal, or approaches the animal, and the animal withdraws and avoids. And so what you can see is both males and females spend less time investigating, and they spend more time avoiding the animal during this juvenile SI test. And when you look at their calcium transients, or the sum of the calcium changes in calcium fluorescence over the course of this interaction, you can see that in susceptible mice, similar to what we saw with our C-FOS assays, susceptible mice seem to have activated LSNT neurons during this period in which they're avoiding the social juvenile target, and this is true both in males and females. So to test whether or not the LSNT neurons were specifically mediating aspects of social behavior, we injected either HM3D or HM4D dreads to activate or inhibit the LSNT neurons. And we were able to modulate both social investigation, social avoidance, as well as conditioned place preference for those juvenile social targets. Let's see. So ultimately what we've found is that activation of these LSNT neurons in the lateral septum are both necessary and sufficient for the induction of social avoidance behavior, as well as reduced social reward. It's interesting to note that when we took resilient mice and did the opposite manipulation, we were able to elicit a susceptible profile based on just manipulating the activity of the LSNT cells. So we had this amazing data set from iDISCO, and I had a postdoc in the lab, Tony Aubrey, who was very interested in understanding kind of brain-wide dynamics of how areas communicate with themselves. So he built this correlational model to look at areas of the brain, or at least C-FOS puncta within areas of the brain that were correlated with one another, which we thought would give us insight possibly into areas that might be functionally and structurally connected and that might be mediating aspects of this social behavior. And so if you look at the LS, for example, and you compare its correlation to other well-known brain areas shown over here, you can see some interesting profiles. So the ventral striatum, which we group together as the nucleus accumbens and the diagonal band, seem to be negatively correlated, meaning higher C-FOS in the LS correlates with lower C-FOS in the NAC and NDB. This made sense given the fact that these LSNT neurons, we know, are GABAergic and could, in theory, when activated, produce inhibition of downstream effectors. These correlational matrices, though, don't take into account any information about actual structural connectivity, right? These are just simply taking all of the C-FOS data and putting them through the analysis to look at what areas are correlated. Those areas might be, di or trisynaptically connected and somehow indirectly correlated in that way. So we performed input-output tracing using various viral tools to confirm which of these targets were actually functionally connected. These are four brain regions that we found to be connected in an anterograde fashion to the LSNT neurons, so downstream. This is a hypothalamic nucleus, the PVT, the PAG, and then the ventral striatum. And then we also are showing four upstream inputs to the LSNT neurons, such as the infralimbic cortex and the CA1 region of the hippocampus, that are retrogradely connected to the LSNT neurons. So they send inputs into those nerve cells. Roman Duran in the lab, another postdoc in the lab, confirmed at an electrophysiological level that these cells are indeed connected to the ventral striatal region, so both the NAC and NDB. The way he did this was to take Cre-dependent channel rhodopsin, inject it into the the lateral septum of the NT-Cre mice, and then measured evoked inhibitory currents from the inputs to the NDB and NAC. And you can see those evoked currents shown down here. So these cells do indeed evoke inhibitory tone coming from the LSNT neurons directly onto cells within the NDB and the NAC. And last but not least, this is very preliminary. We long conducted this study, I think the week before we went into shutdown mode. So he injected, again, channel rhodopsin directly into the LSNT CRE line, and then light evoked inputs to the NAC NDB region during social investigation. And this circuit seems to, when activated, inhibit social investigation time. So we're further probing this circuit to see what its role is in other aspects of social reward as measured in the CPP task. And so this is kind of our ongoing effort. Long has undertaken kind of a Herculean task to probe each of these circuits in and out of the LSNT to see what their roles are in distinct aspects of social behavior. I think he's gotten through 4 or 5 of them already, and we're parsing through that data to see what role our initial belief based on the preliminary data is that each of these LS connected networks may play a different role in an aspect of social behavior that we're measuring. For example, the accumbens seems to be regulating social investigation time, and we predict that it's going to do so by also regulating social reward. Whereas other structures may be playing roles in other aspects of the interaction, such as social avoidance. We saw no evidence that the NAC itself mediates social avoidance behavior. And so we're thinking that some of these other output structures may more directly regulate it. And then ultimately what we'd like to do is rebuild, using C-FOS mapping and chemogenetics, we'd like to rebuild the LSNT network during social interaction with various parameters of activation and inhibition. So for example, inhibiting the LSNT node and then seeing how that regulates brain-wide C-FOS patterns down and upstream of this structure. I think this will give us a better idea of how the brain is integrating information during social interaction. I'd also just like to highlight a few of the studies that I didn't get to talk about in the interest of time. As I mentioned, we've now got a great model for female aggression that's been spearheaded by Tony and Joey in the lab. This is another form, it's an ethologically form of territorial aggression where females are cohabitating with a male. We happen to use castrated males to reduce the confound of pregnancy, but it works with both intact and castrated males. Under these circumstances, we see similar degree or similar amounts of aggression towards intruding females. We've also been working with some automated behavioral analysis. This is work that's being led by Joey and many others in the lab. This is with a program that was developed a couple of years ago called Deep Lab Cuts, which uses artificial intelligence to automate the behavioral analysis. We're also now implementing a new program called Simba, which was developed by Sam Golden, now who used to be in my lab and is now an assistant professor at Washington University. And these tools are extremely powerful. They're allowing us to track very discrete social behaviors during these interactions in both aggressive, non-aggressive mice, as well as the intruder. And it's allowing us to understand aspects of behavior with far greater precision. Again, I mentioned we're doing a lot of whole brain analysis, and so we're continuing our efforts at trying to understand how specific nodes within the brain, for example, LSNT neurons, might be mediating brain-wide dynamics during social interaction. Roman had started this study and had to cut it short due to COVID, but hopefully we're going to ramp that back up very soon. He's very interested in understanding decision-making processes that occur during these social interactions. So in other words, what makes an animal attack versus not attack, and why do some individuals seem to enjoy that interaction, whereas others do not? So for these studies, he's utilizing well-established operant models to parse circuits that are activated during these important decision-making processes. Catherine Leclerc has been studying social hierarchy, so the impact of your place in society on a lot of these phenotypes of social behavior, and she's presented her work at quite a number of venues that I'm sure a lot of you have seen. And then last but not least, my lab is also quite interested in the immune system and understanding neuroimmune interactions. And so I didn't get to talk at all about those studies, but Florin, Kenny, Carrie, Leona, and Yusuke are all doing incredible work on understanding how the immune system, particularly the peripheral immune system, interfaces with the brain during various disease states. And then last but not least, of course, I'd like to thank the lab, my current lab, who have been amazing during this really challenging time. I can see all of them are chomping at the bit to get back into the lab and start doing their experiments again. And we're quite happy to see that we might be doing that soon. I'd also like to thank past members of the lab, and including Megan Flanagan and Sam Golden in particular, who really led most of the studies on aggression, as well as the collaborators, most of which I've mentioned throughout the course of the talk. So I am done, and if anybody has any questions or comments, I'm happy to answer them.
That's great, Scott. Thank you.
Let's see. There's one question from Dan. Do you think giving oxytocin or an agonist to the social defeat mice would you would expect to see a return to normal social reward? I would predict it. There's been some work on the oxytocin systems. There does seem, this was work I think from Larry Young's lab, there does seem to be dysfunction within oxytocin receptor function in the ventral striatum of susceptible mice. So it would make a lot of sense that by restoring normal oxytocin function in a region like the accumbens might normalize social reward. And then we've got a question from Mike Barry. When comparing areas with possible links to septum, it appears that the C-FOS activity was differentially correlated in the susceptible mice versus the resilient mice. Yes, if I understand that question correctly. So areas that were correlated in susceptible mice were oftentimes oppositely correlated in resilient mice. Let me know, Mike, if that wasn't what your question was, and I'd be happy to answer again.
Scott, this is Eric. That was a great talk, and it's great to see how the work has been developing, really very creative and novel.
Thanks, Eric.
We talked about this before, and the notion of whether an OX2 antagonist might be explored for different forms of aggression in humans. I think J&J has an OX2 antagonist.
Yeah. And
have there been studies in aggression models like Alzheimer's aggression or something like that? So yeah, so that's a great question. So J&J does have a compound. Megan and I have a use patent out for its use. We've tried to get them engaged, and they have not shown them enough interest. Actually, surprisingly, where we have seen interest is in the veterinary community for treatment of aggressive dogs, for example, or pets in general. And so that's an area I think that could be interesting and developed a bit further. The J&J compound, I think we tested it, and in our models, it showed anti-aggressive features. The issue with Alzheimer's disease, and this is an area that we've been trying to build and develop, is twofold. One is that any compound that causes any degree of sedation is going to be tricky in an Alzheimer's cohort because it'll make their symptoms worse. And so I think finding the right dose of an OX2 antagonist will be tricky in that population, but not impossible. The other problem is we haven't really been able to find models that in mice recapitulate that increased irritability and aggression that you see in AD patients. We've tested with Joe Castellano's group a number of of established mouse models, and we simply don't see it, although it has been reported in the literature. So it's something we're interested in, but we haven't quite gotten there yet with the models. I thought I saw maybe one other comment, or a few other comments. So this is from Abba. Would you think if the aggressive mice were exposed to stress or trauma, that would alter their behaviors? I was thinking in line with trauma exposure increasing aggressive behavior.
So the answer is yes to both of those questions. So in the immediate term, if you pair the aggressive mice with a bigger bully that attacks and subordinates him, they actually show neural signatures of subordination, similar to what we see in our socially defeated mice. And on the converse side of that, we actually see a reduction in aggression though in the acute phase of that interaction. But if you chronically socially defeat mice during the juvenile period, so this is when they're quite young, and let them develop until adulthood, those mice that were chronically subordinated as juveniles show a very strong increase in aggressive behavior as adults. So suggesting that maybe those kids that get bullied chronically as kids might show or elicit aggression and violence as adults. Okay, so C-FOS does not label all the neurons. If you use other activity like phosphoribosomal proteins, maybe it will help to identify some unknown neurons. Absolutely. You know, especially with iDISCO, not only does it not label all neurons, it labels a whole bunch of all non-neuronal cells. And so what we get from those whole brain patterns is just a gross understanding of how particular brain areas, not just neurons, are activated or inhibited. But we're always very interested in additional tools. And so we'll probably come talk to you about those ribosomal assays. Siva, that's a really good suggestion. Rita, I was intrigued about the IL and CA1 inputs to the septum. What are your thoughts about extinction in this model? It's a great question. Daniela Schiller and Roger and I, years ago, had talked about this, and we'd love to re-engage at some point and maybe develop a project. But maybe what we were measuring in our social defeat model was more related to PTSD in the sense that they were going to be inherently unable to extinguish to this form of learning, right? That social avoidance or social reward was such a profoundly strengthened behavior because of the trauma. And those targets, both IL-1 and CA1, would make excellent targets to test those theories. I don't have a great answer as to whether or not they do, but I think your suggestion is a great one. And then let's see, Regis. Are non-aggressive social behaviors affected by manipulation of the lateral hypothalamus? From the videos you showed in the first half of your talk, the inhibition looked like it was not interested in the other mouse at all. I wonder if you're making the target mouse socially invisible or reducing aggression versus reducing aggression. Yeah, it's a great question. So we never did direct stimulations within the lateral hypothalamus. We only stimulated inputs to them. And it certainly is possible. We did measure, I didn't show the data, we did measure non-aggressive social interactions. So we have a whole host of things that we measure, which is why these automated behavioral analyses are going to be a godsend when we don't have to sit and watch videos for days on end. But we measure a whole host of non-aggressive social interactions, and we did not see any significant perturbation of those non-aggressive social interactions by manipulations of the inputs. But that's, again, not to say that, that the animal is less interested. Maybe not invisible, but maybe that they're just less interested in interacting socially. Something we'll have to do in far greater detail for future studies. And then the last one is from Estrella. Have you assessed if these effects may depend on age, learning mechanisms? Great question. They absolutely do depend at some level on age. So, you know, very young mice, for example, we don't see a lot of evidence for them engaging in this type of aggressive behavior. They do, however, engage in aspects of play behavior, some of which can be rough and aggressive in nature. And so we haven't teased that apart yet. As you move through adulthood, between early and middle adulthood, you start to see an increase in these types of overt aggressive behaviors, at least in males. We have not yet done equivalent studies in females. And what's interesting is there's a couple of critical life events that we find mediate level of intensity of aggression. Obviously, the first is puberty. The second is later stage adult sexual experience. So we really don't unmask the full repertoire of aggressive behaviors until these aggressive mice, or until these mice have been cohabitated and allowed to breed with a female. And then the last phase is as these animals get really old, like somewhere in the range of 9 to 11 months, you see a really dramatic drop off of aggression as they age, which we would predict is probably related to testosterone levels dropping off. So I think that's it on the chat line. If anybody has any other questions, or hopefully we'll chat in person soon.
Yeah, no, that's great, Scott. Thank you again, and thank you everyone for joining us for this fabulous series.